Okay, welcome everyone. This is Caitlin from BBMI ERIC. We're really happy to have this webinar today with Mary Wong to talk about biobanks and registries. We found that we can be sometimes in our own bubbles and think that everybody understands what we're talking about when we talk about biobanks and registries, and realized in conversations that there's a lot of confusion around these terms, and getting some basic understanding would be really important for all of our stakeholders. So we have a lot of people from different organizations joining today, and if at any time you have questions, you can raise your hand. Everybody's automatically muted, but you can raise your hand on the little hands up button next to your name, and you can also type your questions in the questions area at the bottom. At the end, we have two very short poll questions, so you'll be able to answer them right on your screen, and we'll get live feedback for that. So Mary Wong is the Scientific Partnership Manager at Fondazione Telethon in Italy. There, she's also the Biobank Program Manager, where she advocates for rare disease biobanking and gives strategic support for the Telethon network of genetic biobanks. Mary is also working on the European Joint Program on Rare Diseases. She leads the Biobank training there and surveys the rare disease community on their research needs. She also worked on the project RD Connect until 2018. And she's in the Euro Biobank Network Secretariat. She contributes to IRD, IRC, planning of a global roadmap, and the ICPERMed Executive Committee. So Mary can answer hopefully most of your questions with her broad experience. So Mary, I'll hand it over to you. And again, if people have questions along the way, just
Hi, this is Mary. So thank you for inviting me to give this webinar. So I will, in this webinar, explain to you, try to illustrate a little bit what are biobanks, what are research biobanks and registries, because I do often, when I go around and in meetings and conferences and also talking to patients, they're often referred together. But so it's useful to just also to remember what are they, what are we referring to when we're talking about these resources. And so this is the purpose of my talk, to see next page, to go over some basics in a way. So it's just to say what is a biobank and what is a registry, and then going to what sort of research do they support and how do they operate, and giving you some examples of that. And then just the last part to also giving you a contrast a little bit between these two resources to see what are the similarities or differences and how they might work in synergy. Okay, so I think I heard now most of, or many of you are coming from a biobank, but so this is my favorite definition of a biobank. So biobank is an organized collection of human biological material and the associated information data stored for one or more research purposes. And so this is really, so you have the biobank collecting samples from different sources, and you process and you storage, and then you have multiple distribution. This part is very crucial. So biobank is not just storing, it's really for distributing and for multiple research. And so this definition comes from this article. What are the different types of biobanks, especially for research biobanks? So they are, as a whole, there are several minor definitions of the different groups, but these on the whole are the main types. So they're either disease-based, so they collect biological samples from patients from specific diseases or carriers, and sometimes also the healthy controls. For instance, here I grabbed a few logos from Biobank. on psoriasis, on cancer, and this is the one on the heart. So they are disease-based. And then there are the other types which are cohort-based. So these could be longitudinal cohort or isolated geographical regions. For instance, the lifelines, which I heard last time from a BBMR Eric webinar. So there, the lifelines in Groningen are a type of example of the longitudinal, actually also isolated, well, not isolated, but a geographical from the north of Netherlands. And Trentino Biobank, so that's an area in a region in Italy, in the Alps. And there are also other examples of the Finnish, the twins. And so you can, so they don't have a specific disease. So these are just containing biobanks containing samples from a subset of population, limited by region or ethnics, and so coming from that focus. So these are the two main types of research biobanks. So biobanks, they may differ from each other in size. So some are much smaller, especially the ones that I know more about. So the rare disease biobanks, they, because of also the patient rarity, the biobanks tend to be quite small. And so they can range from holding a few hundreds of biological samples to millions of, as a populational one, millions of samples. So they can really vary in size. They can cover different geographical level of coverage. So some are regional, some are only national, some are maybe international. They have different also kind of ownership or hosting institution. They might be in hospitals, or they might be coming from universities. They might be of a non-profit or foundation, or they may be also even hosted inside companies. So biological samples being stored, there are many types. So here I've only listed, let's say, the often cited ones, so from blood, from urine to tissues, and also its derivatives. So from blood, you can have plasma, serum, DNA, the lymphocytes, PBMC, etc. So many tissue types that are being stored in biobanks, and some biobanks may only collect DNA, or some might only be focusing on cells. So they have a different repertoire. You can see these differences. And then some biobanks also provide additional services. So there might be also a difference in that. Then some of them might also be able to perform some experiments, or they provide validation on quality of samples, or they can have additional derivatives coming from the original tissues, or for instance, the juiced pluripotent stem cells from urine or fibroblasts. So not all biobanks can have these kind of technologies in the laboratories, or other services on request. So although we might think biobanks as a whole, as a group, but within them, they actually can be very different in terms of the services that they provide. Next. So this is the central biobank workflow. So how do the biobanks operate? So this is, in general, they all work in this way. So how the samples are coming in, they're coming from the patients and participants with a signed informed consent. And then the sample is then banked, stored in the biobank with its quality assurances and standard operating procedures. On the other side, so on the external side, then the whole purpose, as we defined in my first slide, is for research purposes. So the researchers then can request access to these biological samples, and this often happens with a web form, or sometimes via an email. So they can write a little request form or little for their projects to have a certain number of samples, or to set up a prospective collection. And then these samples then can, once agreed, then they can be sent to the researchers for experiments. And this is usually accompanied by a material transfer agreement signed between the researchers institute and the biobank themselves. So biobanks maybe seem simple in that previous slide, so sample coming in, sample going out, but actually there's a whole lot of areas that needs to look at. And so these are really, biobanks are really professional research infrastructures. And so besides brokering access for researchers who might not have materials, they need to be able to warrant the quality of the biological material over time. And so the processing on the quality and the SOPs are very important. It needs to be able to warranty that so that it can have confidence for the research reproducibility for also future technology. So not just collecting for the technology now being available, but also thinking of possible future technologies. And it also manages the legal and ethical issues, as you can imagine, coming from the informed consent, and also talking about many of discussions more recently about the return of results and what that means to the patients. And so you can see here on the right, so like 360 degrees of biobanks, and it also needs to have an outreach on make sure that the researchers or the community are aware of the biobanks. It has a website, how to make its catalog available and findable to the community. And on this side, the VMI, Eric, has been working quite a lot in this past few years on the directory and the negotiator, I think. Biobanks needs to be operating with the long-term sustainability in mind, and so it needs to also have a good business plan for continuity and make sure that its service is provided professionally. So you may have requests coming in and you cannot just leave it there and reply only 2 months later. And so you need to operate in a professional way, and requests coming in, and you have to take care of them, and also making sure that the samples gets shipped out in due time. So I think it's quite simple, but they're actually very professional organizations running these biobanks. So what kind of research do they support? So I'm now going to bring you 2 examples from the Italian, the Telephone Network of Genetic Biobanks. So this is a story actually long ago now, in 1999. So there was a family in Genoa, in Italy, where babies died very early in their infancy due to an immune deficiency disorder. And so after genetic counseling, they agreed to deposit biological samples in a biobank. And then in 2001, so scientists in the USA, so the samples were banked in Italy, but the scientists in the USA were able to find and request these samples and then actually identify the mutation in the causative gene, and also identify the gene as the FOXP3 that was underlying these IPEX immunodeficiency. And then in 2006, and also the samples were used to develop validation for the genetic test for prenatal diagnosis. And then 11, so the samples are also used for pathophysiology studies to try to understand how the molecular pathways and how it actually works in this gene. And so you can see samples was banked, and the materials can be used in different types of research, from gene discovery to prenatal testing kits and to understanding of disease. And so this is a really nice example of how samples from one place can be made available to researchers in different parts of the world, and also different types of research. Those who are familiar with in the immunology field, you might be aware that the FOXP3 then is an important marker of regulatory T cells, and which then also opened up a big field of study immune regulation, and also more recently in the cell therapy field. And this is another case. So besides looking at the research articles, it's also really important to remember these are families and real people behind with stories. And so this is, I think biobanks also are there to provide answers and hope for families. So this is a story of Nicola and Rafaela. So they are sisters in their 20s. And so since their birth, they had developed from 3 months of age. They showed symptoms of tremors and then later on developed intellectual disability with heart problems. The family had no diagnosis for over 20 years, and they went around. And then eventually, after depositing their DNA at the biobank, so this case in the south of Italy, they discovered so now with the technology which became more and more prevalent on the West, whole exome sequencing, they identified a mutation to this GNB5 gene, and actually it's a novel mutation, so it's a novel disease. And that sort of snowballed into also identifying other families in the world. So in this paper in 2016, there are also families coming from, I think, Turkey, and Netherlands, and Saudi Arabia, and different families from the world. And so for Nicola and Rafaela's family, they eventually also received their diagnosis. So this was in 16, and then more recently, so actually this year, the laboratory in the biobanks are also continuing the work to understand this disease, and also there are other examples of generating iPS cell lines, so disease models using patient specific pathogenetic variant to make iPS cell lines. So this story is actually also, you can read it on the BBMI website with a link on the top. And so other than this is of course also research, but also just wanted to remind everyone the people behind the patients who are waiting for an answer. And so now I move to registries. So what are registries? This is the EMA definition of a registry, so an organized system that uses observational methods to collect information, collect uniform data on a patient population defined by a particular disease, exposure, or condition, which is followed over time. So these are a few key points here. So they have a particular condition and it's followed over time. And then the patient disease registries can be established by public organizations, from academia, investigators, medical associations, hospitals, but also patients. So this is something that is more and more we're hearing that patient organizations are also very keen in looking into setting up patient registries. These registries, they are set up, they may have different objectives. So these are some examples, such as to describe the natural history of a disease or a disorder. So how does a disease progress over time? How does it develop? And also they can be used to monitor the efficacy, effectiveness, or the safety of a treatment. So with a therapy already in place, and to describe the impact of disease on patients' health and quality of life, and also to often used to identify patients suitable for treatments or for clinical trials. And registries usually are given through questionnaires, and they typically have these categories of data collected. So there's a baseline information about the subject. So you have patient demographic information, its consent, the consent that took been registered, date of birth, etc., etc. It has asks about the medical history of the patient, also not just about the disease itself, but they may also ask, for instance, whether the patient has asthma, or if they sleep well, or what are sort of and then also more patient characteristics like so quality of life questions, occupation, diet, lifestyles. Medical history is maybe more clinical information, the kind of different maybe for genetic mutations, variants, and also different laboratory tests information may be also registered in this register. So here I just took an example of the UK FSHD's facioscapulohumeral dystrophy. It's a genetic disease patient registry. So here you can see example of questions that are asked. So whether you have genetic information, so actually mutation for your disease, what kind of diagnosis you have, what kind of symptoms you may have, and you can take different things. So this is example of the questions and the data that go into a registry. How are the information gathered then? So this is the registry workflow comparing to the biobank workflow. So this is the sort of traditional way how the data is collected from patients. So you might have the subject, the patient themselves, where they look at the privacy, they look at the consent, and then they can look at start filling the survey, the questionnaires of the registries, and then they may go to the clinical visit, where then the data filled then is then validated with the physician. And in this particular registry taken from the Italian neuromuscular network, so there's also sometimes a secondary validation step or cleaning from a curator from the scientific office of the registry. And so you can see how the data is sort of coming in and validating each stage. And then so at the end you have this database where then they can be used and then regulated by a registry governance, for also then you can be used for publication and for recruitment of patients, etc. So that was the sort of more traditional way of data entry for registries. So that's what I described. So maybe investigators, clinicians from different hospitals, they may use a web form, and then they have a registry database set up, and with sometimes patient involvement, sometimes not. And so, but now more and more registries are designed with patient-centered approach, where patients and families can fill in the questionnaire also through, you can see in this case also through mobile phone apps, where they can just enter the data more quickly, and also perhaps getting some of the results back and reading what is being available. And so on the one side, you have the physicians also be able to see entering the information from the clinics, from the laboratory into the registry, and so it's more collaborative and it's more patient-centered. So this way, it's also helps the retention and the completeness of the registry, where patients are being more involved. So sometimes for longitudinal study, maybe for a patient not maybe need to be followed on for many years. So having something really accessible, not having to visit the clinic every time, is something that helps the process. So what are then the elements to think about when you perhaps set up a registry? So similarly with the biobanks, so you need to think about which patient population you would like to include. What are the kind of time that are important for the particular disease? What kind of data you want to record? So there's a core data element, something that examples that I've given before. How do you standardize those terminologies, those data elements? And how do you make sure that the data are registered accurately and on timely basis? So if some event take place for a disease and you don't want to just think about it 6 months later on. So they have to be able to have fit for data or privacy requirements, as well as having a good governance. So access policy as well, and ownership, and these are things that all need to be considered for a registry. And what kind of research does it support? So registries are, so this is a sort of typical research drug development pipeline, from discovery to market authorization and also post-authorization. So registries can be are useful in many of these stages. So in the discovery part, you can using the natural history studies, so you can understand how disease are progressing and how does it change over time. In the preclinical stage, you will use the registries to also look at what will be to design your trial, to design your inclusion criteria for your trial, and also, of course, to identify suitable to recruit patients for trials. So here I also put some little icons to see who might be interested in these stages. So here in the early stage, it might be the researchers trying to understand the disease, the clinicians trying to set up a trial, the industry maybe also to really have the clinical trial going, looking for the patients. And then so later on, when you actually have a trial or a therapy coming out, then also, so the regulatory authority also looking at that to help support, for instance, the reimbursement decisions, and also the after authorization. So in the real world, for monitoring how the drugs are safe, to collect evidence of the effectiveness of the therapy, etc. So really also in different stages of the therapy development. Sorry, excuse me. Oop. And so here I'm giving you some examples of registries being in use in what I just showed you now. So here you can see the first one is I just found one of from the cancer registry, or neurofibromatosis. So this American, or actually international registry, where they, in this article, they reported in a 3-year period, this registry has been used for different types of studies. So you can see on the little table on the top, observational studies or natural history studies, for drug phase 2 studies. So different types of examples, and also, for instance, also quality of life questionnaires development. And so you can see this one as an example of one registry being used. And also the second one is a observational registry for monitoring the safety and effectiveness of gene therapy for 15 years. And this is actually an EMA regulatory requirement of an approved therapy. Okay, so then now I've shown you some basics on registries and biobanks. So here I can see that registries and biobanks are really two sides of the same coin. They are both really valuable for research, and both of them collect resources, so data and biological samples. They both require good data management, quality management, and also governance for their operations. Both of them also involve multiple stakeholders, patients, clinicians, and researchers. And also now existing, there are international, national standards for their operation, and also there are many, many networks now for both, and both of them need to operate in a sustainable method. So here I have a table, so it's non-exhaustive, just to compare the biobanks and registries. So the main resource of a biobank are the biological samples, and they do collect also clinical data, some associated data, but usually not in an exhaustive way compared to the registry, where also in the registry also they contain more different types of also quality of life, medical history kind of information. They support different types of research, so mostly translational, and the biobanks was registry. They're often supporting clinical trials and design and understanding of disease. And they similarly have biobanks have focused on the disease conditions or cohorts. Registries tend not to have cohort registries in a way like without having a disease or condition. So this is something slightly different between biobank and registry. And so here some of the operational aspects are both very pretty much the same. You can see that they both require good standards and good operating procedures. So now I'm coming to talking about the synergy. So I showed a few examples of the biobanks and what kind of research that they do, and then the registry. So here the little database icon. Are they working together? What are the values of having them working in synergy? And this is a particular example, I think, where, for instance, studies looking to identify or validate biomarkers are of value when biobanks and registries and actually work together, because they enrich each other's data, and in the end they're about the same patients, operating with the patients in mind to accelerate and facilitate research. This is so for instance, in a biomarker study, you want to validate how to pick up biomarkers that change in a disease over time. So maybe in the initial phase, maybe you have a low reading of something or expression of a low biomarker, and then that might change over time, or it might change something you want to pick up over the course of treatment. And perhaps some data are recorded in the registry as progressions or as a part of natural history studies. But if you have the biological, but within itself, you cannot have biomarkers. Whereas on the biobank side, having the biological samples, but without all those associated quality of life data or the medical history, then they're not as valuable as they can be. So having these two resources together, or in interoperable way that you can call data information from both sides, then they would make such studies extremely valuable. And so this is the importance also now, I think now examples and studies of biobanks and registries trying to integrate more with each other. So that this is a way to support research. Okay, I just have a slide here. I want to recall patient involvement. So both of these, the biobanks and the registries, they have a strong patient focus. And so I just want to have a slide here to say patients can be and are involved in many of these aspects already. For instance, patient can be part of the governance of a biobank or registry, and there are examples of that. They can drive the creation of samples, collections, and the databases, and also driving the collaborations between them, because sometimes they are the part that's in the middle of the two circles. So they know where their samples might be, or they might know that they are putting their questionnaires, the registries in a different place. So they can also really drive the networking and collaborations. And it's also for ways to have a social engagement, also to explain to the community what are the values of biobank registries in research. So yeah, now the biobanks and registries are paying more and more attention to, or designing themselves to be more patient-centric, and I think this is a great direction. I want to have one more slide, I think. So that's a summary slide. And so I've hopefully shown you that the biobanks and registries are fundamental resources for research. They cover different parts of the drug development pipeline and understanding of disease. They share many common features, and certainly, especially in the operational front. Yeah, so they support complementary. So I wanted to emphasize that. So they don't, you might think, oh, registering biobanks, they support the same research, but no, they actually support complementary types of research activities, and they should be made interoperable to each other. And that really would create more value and create synergies together. And that is the end of my presentation.
Thank you very much, Mary. This was very interesting. Does anybody have any question, or you can raise your hand, or you can write it in the question space? Or if you have a question after this webinar, you can also, of course, send it to us and we will answer as soon as possible. Very good. So we have 2 poll questions, and may I kindly ask you to answer these? So this is the first one. So the poll is open. This is anonymous, and we will really appreciate that you answer them. So I will give 1 minute of time.
I don't actually see a poll myself. Maybe I have to stop sharing my screen.
The participants, I think they see it, Mary.
Okay. All right. Okay.
But as you are in this side of as a panelist.
Right. Okay.
So the question is, do you understand better the difference between biobanks and registries after attending this webinar? So I have here a percentage that says no. In case that, please, you can ask something to Mary. Or again, you can send it to us and we will answer you as soon as possible. I can see here a question. So Mary, the question is, are the sample and data stored into the biobank fully anonymized? Ruggero Di Maulo is asking this.
So data are not anonymized because yeah, so what the data, the biobanks or the registry themselves, they can always refer back to the patient in case of particular health information that come out of research. So within the biobanks, they're not anonymized. They may be coded or pseudonymized when they are stored, but they are not anonymized within the biobanks. When they distribute them, then they would be anonymized. So they keep the information within themselves, but not for management purposes. Does that answer your question?
Yes, he said thank you.
Okay.
Very good. So let's go to the second. Let's see. So we close this. And now, have you used a registry in the past? So yes or no. We have 50-50%. Oh, now 75. Oh, it's moving. So 23% yes, 74 no. Okay, now it's changing, 26 to 74. Most of you haven't used one. That is a very interesting result. Very good. So I'll give it 20 seconds more. And please feel free to ask something. Very good. Okay, so you will receive the it's possible to download the presentation that Mary gave us today. You can send us your questions, doubts about this topic. And thank you very much, Mary, for this. It was very, very interesting. And thank you to all the attendees for your attention. I wish you a very nice afternoon. See you next time.
Thank you. Bye-bye.